what you have listened last 3 days. So this is more of innovation opportunities in COVID pandemic mitigation. Being a biologist, I will try to encapsulate all these aspects through the biological journey in the COVID-19 pandemic. So if we browse through the COVID pandemic fact, every day we are looking at our mobile and looking at the total number of cases in the globally in India as well as local in our Ahmedabad. So if you look at the global distribution of cases, India is ranking around 11 with a 2.09% of contribution. And if we see the total number of cases, it's around 1 lakh in India, and it is around 12,000. This is the data of yesterday night. So looking at these ever rising cases, it is really a fearful situation in all of us, our mind. Now, let's go through the origin, transmission, and characteristic of this COVID-19. So if you look at, it is a SARS-CoV-2 virus, which is phylogenetically and genomically similar to the earlier SARS viruses like bat CoV as well as in MERS, with a primary host in bat and which is transmitted to human host through the food consumption source. And it is also speculated that it may have some intermediate host to which the human being is in direct contact. And now we are in the phase of human to human transmission. And we are struggling for various therapeutic strategies, what to do in this situation. So let me quickly take you through the organization of the genome of this COVID-19. If you look at the whole genome, it is around 30 kilobase pair. And to be precise, it is 29,800 odd base pairs. And the whole genome architecture is divided into 3 major segments like ORF1, ORF2, ORF1b, and the various structural protein like spike, extracellular membrane, and nucleocapsid proteins. And if we say in a single line, we can say that COVID-19 is an enveloped single positive stranded RNA, which is 29,811 base pair long. And as in these last 3 days, we have listened a lot about the structure and all this, so I'm not going to the detail. But interestingly, if you look at the life cycle of coronavirus, from entry to exit, there are lots of opportunity. So all the red T marks, if you see, these are the possible opportunities where we can intervene. So this virus detects entry into the human cell, specifically the epithelial cells of nasal sinuses as well as in the lung epithelial cells. It can enter through either endocytosis, forming an endosome, which may be clathrin mediated or independent of that, or it can fuse with the membrane where it is interacting with ACE2 and TMPRSS2 receptors, and which makes them to fuse with the membrane, and ultimately the nuclear content is poured inside our cytoplasm. And once this RNA is put in our cytoplasm, immediately first copy of that is being translated and it forms the PP1A and PP1B fragment of that. And ultimately, these give rise to various non-structural proteins. And these PP1A and PP1B is fragmented into 2, which gives rise to helicase and RDRP, that is ribonuclease dependent the ribonuclease, and these helps in replication of the viral RNA into multiple copies. At the same time, the viral structural proteins like spike protein, membrane protein, envelope protein, these are also synthesized at the ribosomes located on the surface of endoplasmic reticulum, and ultimately these are shaded off and in the form of the Golgi complex, and the RNA that is being duplicated along with the nucleocapsid is packaged inside this, and ultimately new virions are made, and they are thrown out of the cell through exocytosis. And these are produced in thousands. So when again these new virions are capable of infecting new cells, and that's how the whole cycle of coronavirus keeps on going. So now if you look at the red T, where you can have therapeutic intervention, capsid protein at the spike protein, or it can be inhibiting the various enzymes, those are involved in binding, or the enzyme receptors which are involved in binding to the viruses. Or it can be at the enzymatic level, which these are the lysosomal enzymes which cleaves these intact viral protein so that the RNA is released into the cytoplasm. So either at this level or during the replication of the viral RNA, so at the RDRP. So a lot of work is being done in this. And many of the antiviral drugs which are currently being used are focusing on this RDRP. Few of the natural compounds have shown, like resveratrol, which can inhibit the organization of the genomic RNA and the nucleocapsid, so that a properly folded RNA is not made and further virulence of the virus is not there. Now there are several questions in our mind that how this presence of virus is being tested in human being. So you must have heard about that whenever we develop fever or some symptoms, the physician will immediately ask us to go for real-time PCR-based testing. So let's quickly go to that. So when you go to the physician, he will take a nasal swab right deep inside your nostril and try to take cells around your nasal epithelium, and they are immediately dipped in viral transport medium. So that keeps the viruses intact as well as live. So now when the sample reaches to lab, the lab people, what they do, they isolate the viral RNA using various RNA extraction protocols. And these extraction protocols can be manual or automated depending on the lab facility. Ultimately, what you get is a pure copy of viral RNA. And this viral RNA is reverse transcribed using reverse transcriptase into a cDNA. And then this cDNA is further amplified using real-time PCR. And in this, what they use is gene-specific primers. So usually two gene-specific primers are used for detection. ICMR has suggested RDRP and ORF gene as the two targets. And these are the sequences of that, forward and reverse sequence, and there is a probe. So probe is a unique signature sequence in between the total amplicon, and so that when the DNA polymerase is extending the primer, and when it reaches the probe, it immediately hydrolyzes the probe. So these are hydrolysis type of probe, in which when the probe and quencher are in close proximity, there is no fluorescence. But as the DNA polymerase, it digests this, the dye is released, or it is now away from the quencher, and you will get some fluorescence. So every time, whenever there is a duplication of the copy, there is a release of fluorochrome, and that's how you see the increase in fluorescence intensity during the whole process of real time. And these are usually the Q is the quencher and FAM is the dye that is being used. And at the end, what you will get a curve like this. So when a curve is going beyond the threshold limit, what we call as CT, the cycle at which it crosses the threshold. And once it goes out of the threshold limit, we say that this gene expression is positive. So when there is no expression, there will be no fluorescence signal. So if both the genes are positive, RDRP and ORF1, we call that as a positive test result. But either of them, if it is not coming out positive within the cycle of 20 to 30, then we may have to repeat the test. Or if both are negative, we can call them as negative. Now there are some quality control issues, like whether the sample has been extracted properly, right? So there are chances of error at the point of extraction. There are chances in the error during the process of replication, like in the PCR machine, or there are chances of DNA contamination, RNA contamination. So when you are processing number of samples, multiple samples, there are chances that RNA from one may come out as a droplet, and it immediately goes into the well, adjacent well, and it may show positivity. So all these quality control issues are taken care during the process of real-time PCR. And that's the reason you require trained manpower for performing this. Another type of test, which is called as antibody-based test or serological test, and in short, it is called as IgG, IgM test. So in that case, what you do, we collect a blood sample, serum is separated, and a drop of serum is put in a lateral flow assay, or we call it as rapid dry test. So this serum, it takes along with that antigen, which is coated with gold beads. And when it passes through this, through capillary action, there are two lines. First one is IgM and the second one is IgG. So the antibody which is specific for this antigen are pre-coated on those lines. And when these blood sample along with the antibody bound to the antigen, when they goes and bind to respective, either it is IgG or it is IgM, then at the end you will get a signal like this. So if there is only one, that means there is a control line. So that control line shows that the test is performed correctly. So if there is only one line, it's negative test. If you get, along with control, if you get the line matching the IgM, so it is called as IgM positive. If you get a second line, that is control plus G, it is called as IgG positive. Whereas if you get three bands, it is called as both G and M positive. So what is the significance of this G and M? If you look at the cycle of our immune response, you take this 0 day as the onset of your infection, and as the viral load keeps on increasing and up around 2 weeks, it is at the peak, and gradually it goes down and subside at the 4th week. IgM detecting after week 1 and it reaches to around 2 and it goes down to base level in 3rd week. Whereas IgG appears from 2nd week and it keeps on circulation for a longer time. So if you get only IgG, that means you have got past infection and possibly you are in the 3rd to 4th week of infection or beyond 4th week. If you get both G and M, that means you are somewhere in between 2nd to 3rd week. And that's the reason you have got both G and M. And if you have only M, that means it is in the early phase of infection, just a week long or between week 1 to week 2. So that's the whole calculation. So if you look at the signs and symptom of the disease and the diagnosis, like the most common symptom, as we all know, fever, dry cough, tiredness, all those. And usually the fever is measured currently through thermometers, and to avoid the person-to-person contamination or cross infection, we are using contactless thermometers. So a lot of business opportunity arises, and you can see hundreds of different vendors coming in the market with various types of contact thermometers. To avoid droplet infection from the infected patient to the healthcare worker or the neighbors or the neighboring individuals who is around you, what we wear is a mask, triple layer mask, or if it is a healthcare worker, N95 mask. And it gives us protection from droplet propagation, droplet-based infection. So now a lot of business opportunity sort of by making face shields, coated mask, reusable masks. So there are a lot of opportunity in this segment. Lot more opportunity came when there was a shortage of RT-PCR kits in the market. So it was not possible for a single company to provide the millions of tests that is required across the globe. So ICMR, what they did, they asked for various companies who were in this business to develop the specific RT-PCR kit, and they took up and tested all the QC, QA, and then they authorized several agencies so that everybody can supply in the market and they can meet the requirement. Demand and supply is matched. Routine testing or the rapid testing of antigen or antibody through the IgG, IgM based test are not still in the market, but they are on the edge of the market and they will be launched soon, because they will be useful when you want to assess herd immunity. So when majority of persons are getting infected and then they are getting normalized, and you want to check whether they have proper IgG or IgM in them or not, so that you can recruit them for plasma therapy, for collection of plasma from them. So less common symptoms, body ache, sore throat, diarrhea, conjunctivitis, headache, all those, rashes on skin. But the serious symptoms, which everyone should be aware of and they should immediately rush to hospital, is difficulty in breathing or shortness of breath. So that shows that now your lung is getting infected or that is injured, and that's the reason you are not able to breathe properly. So that may be along with chest pain or pressure in the chest cavity. Many a time loss of speech or movement. Now in this case, usually the patient is subjected to CT scan to look at what is the condition in the lung, whether there is fibrosis, whether there is fluid filled in the lung. So now the question arises. So a lot of opportunities available at this place, because when the patient is in serious symptoms, that means he is heavily loaded with virus, so heavy viral load. So any close encounter or any close proximity makes you susceptible to get the infection. So in that case, any invasive method or any direct examination by a healthcare worker is a risk factor. And two opportunities were there. One was ventilator. So if you have heard, in the initial 1 month, there was a crisis of ventilator across the globe, and all major companies who can make, they started making this in thousands of numbers. So the challenge was how quickly we can make and how affordable we can make, so that it reaches to the masses. Second, which is still a challenge, is can we make a wearable device which can be stuck to the chest wall and that can, sorry, monitor your lung performance, like the breathing rate, the shallow breathing or deep breathing, so, or the lung sound that is being coming out of the chest wall. So such devices are in need. So any person from electronics along with a medical professional may join up, team up, and they can work on these areas. So we usually look at every day, how many people died today, right? And this is increasing daily, and possibly the death rate was highest in the week of April 13 to 19. So that was the peak week of death. So around 7,500 individuals were dying every day. The highest was possibly 8,500 somewhere. And we were panicked. But if you look at the other death rate, how many people die daily, each day, with different other reasons, then you can see around 48,000 with cardiovascular disease, cancer 26,000, respiratory disease 10,000, so many. So now if you compare, possibly the death rate is not that panic. But the situation is panic because it is actively spreading to anyone. So it was not sparing anyone, whether it is rich, whether it is poor, whether it is a hawker, whether it is a caretaker, it has not spared anyone. And that's the reason we are panicked. And there were quick observations that around 73% of the people who died were male with median age of 66 years and with some underlying chronic conditions such as heart problem or diabetes. So why do people die of COVID? So first and foremost is lack of immunity, and that's the reason they are susceptible. So around 5% of patients had severe lung injury and or even multi-organ dysfunction during the course of this infection. And that ultimately leads to respiratory failure, cardiac failure, viral sepsis, or cytokine storms, which is produced in response to the lung injury. And the fatality rate is somewhere in between 1 to 3%, and that is variable from country to country. So now the question arises, how to assess the immunity, right? So everyone must be aware, oh, how is my immunity? So if you, this is just a cartoon diagram to depict the immunity protection from an individual. So if you can see, the size of the umbrella and type of umbrella is equivalent to the type of immunity that you have. And when the patient is having infection, he can be asymptomatic, he can be mild, he may lead to moderate, severe, and ultimately fatal. So this is the track in which we have to go. So whether you stop at mild, whether you stop at moderate, whether you stop at severe, or ultimately it can be fatal. So what are the factors that contribute to your immunity? First is your diet and lifestyle habit. That means ultimately we all are living in a oxidative stress situation where the amount of oxidative stress is way beyond our antioxidants capacity. So these antioxidants are present in the form of enzymatic antioxidants like GST, GR, SOD, catalase. So those are there in our body. Or it can be non-enzymatic antioxidants like several vitamins C, D, A, trace minerals, zinc, selenium, and the phytochemicals that we get from fruits and vegetables. Factors affecting immunity may be pre-existing illness like hypertension, diabetes, autoimmune disease, like transplant case, cancer, or HIV. So these are having low immunity. It can also be, now it is presumed that immunity is also associated with genetic and epigenetic makeup. So genetic, when there is a defect somewhere in the, say, mutant SOD or a null type GST or a mutant catalase, then these enzymes are functioning at a lower efficiency as compared to the normal one. Or the epigenetic can be a epigenetic regulation through promoter methylation or some other epigenetic ways of regulating the antioxidants. It's also affected by your sleep and wake up cycle. So if you are not sleeping well, or if your wake up cycle is disturbed, then there are chances that your immune system is also affected. If you took a real view of the whole immune system, we can divide this immunity into innate and acquired, or you can say adaptive also. And this acquired immunity can be in a natural way, like from maternal transfer when we are born, or through infection when we encounter any bacterial or viral pathogens. It can be artificial through the way of vaccination. So vaccination is a active artificial form of immunization, whereas injecting antibodies, what we also say the plasma therapy or the ready-made antibodies against the antigen, COVID antigen, can be an artificial and passive immunity. And all these cells which are responsible for maintaining our immune system are mainly generating from the bone marrow. The bone marrow, having the hematopoietic stem cells, they further differentiate into various progenitor cells, lymphoid and myeloid, and they give rise to all our cell types: monocyte, basophil, eosinophil, neutrophil, platelet, all those. And the lymphoid progenitor give rise to the main fighters like T lymphocytes and B lymphocytes. Ultimately, these B lymphocytes are responsible for producing the required antibodies, and T lymphocytes, along with their helper cell, T regulatory cells, and cytotoxic T cells, they give, or they are the contributor for our adaptive immunity. So if you look at broadly the circulatory system and the lymphatic system, the circulatory system is mainly performing with the pumping mechanism of heart. And that's the reason the blood is constantly flowing throughout the body. But the issue with lymph is that it doesn't have a heart of its own. So the complex network of the whole lymphatic system, they have certain nodes or the regional pumping stations, we can say, but that is only active when you work more or when you exercise. So that's the reason everyone says that be active and do some exercises in the form of physical exercises or in the form of yoga, pranayama, all these, because all these junctions of lymphocytes need to be praised or need to be compressed with the process of your physical activity. And that's how the whole lymphatic system, which carries all your waste from the body, and even that is the route to which your immune cells are traveling. So what is the chance now? One is chemoprevention for COVID-19, right? So if you look at the allopathic people, currently we don't have a good chemopreventive agent. So due to the pandemic panic, what we did is whatever that were available in our arsenal, they tried to repurpose the drug and tried to look at that which drug can be salvaged for either chemoprevention or for treatment of COVID. And you can see hydroxychloroquine, remdesivir, and anti-HIV drugs and few of the antibiotics were tried and tested, but still their efficiency or their treating efficiency is questionable. So still a lot many clinical trials are going on a larger scale. Now, that's the reason people looked at Ayurvedic practitioners. So in Ayurveda, which is a age-long treasure of Indian system of medicine, and that's the reason whole world is looking at us, that can we provide some solutions to this issue. Even Chinese system of medicine tried many of their Chinese herbs, and that worked well. And that's the reason we also adopted, and Ayush become more active. Uh, the support from Honorable Prime Minister. And you can see every day you will be getting some prescription, right? The Ayurvedic prescriptions. That combination of this decoction is good for this. And even people tried dry ginger powder. But later on, Ayush prescribed a decoction with all its formulation, so that people don't rush into all those are being shared in the WhatsApp. In this case, we venture into one formulation that we called it as Immunize. So Immunize is a patented polyherbal formulation from one of our collaborator who is associated with Gujarat University. And we have proposed this for a clinical trial to Ayush, and we are waiting for its approval. So this Immunize is a polyherbal formulation, and it has been tried on various other illness, chronic illness, like viral hepatitis, even HIV. So a lot of the earlier report that prompted us to think on the direction of anti-COVID therapy also. Along with that, Ayush is also prescribing yoga and pranayam for proper lung support. The third wing of Ayush was homeopathy. And one thing that was viral and everyone must have tried is arsenic album, 3 days. And that was the drug which is being prescribed by homeopathic group. So where is the opportunity here? Being a biologist, we need to establish many in vitro models. We need to establish several biomarkers and to validate this claim. So someone, whether it is from allopathy, whether it is from Ayurvedic, or whether it is from homeopathy, if he comes to you with a drug that, can you please test me for whether this is antiviral or not. But currently we don't have an anti-COVID-19 solution in the form of in vitro model, or in the form of in vivo model, or in the form of any other biomarker to try. Because to handle these viruses, these are highly pathogenic viruses, and they transmit through aerosol. So what you require is a BSL-3, BSL-4 type of facility, and that is limited. The availability is limited. So it is only available with very few labs like NIV, National Institute of Virology, and few other labs in the country. So there is a lot more scope to establish the in vitro models and to establish biomarker for this. Other preventive measures, we all are aware of this, right? That wear mask. So what is an appropriate mask then? So people suggest that N95 is required for healthcare providers. Otherwise, for common people, a triple layered mask, which is disposable or reusable, can be preferred. Personal protective equipments. So that is for healthcare providers who are in close proximity of the patients. Laboratory personnel who are involved in diagnosis, they also need this PPE. Now, there is a lot of opportunity that is going on. You will see that hundreds of commercial ads are coming up that we have a PPE with the whole set is 450. Someone will say it is 1250. So lot of innovation in fabric as well as the coating material is there. So those who are onto, or those who have interest in developing fabrics, or those who are working on nanomaterial science or nanoscience, and they come up with some nanoparticle based coating onto that fabric so that it becomes more efficiently antiviral, and if it can be reusable, say 25 watts, 50 watts. There are many claims now. So there are a lot more innovations or opportunity possible in development of PPE as well as in the mask. Social distancing is for common man, and that is the only one thing which can be done without any money, and that is the preventive measure. Hand cleaning with soap or sanitizer. So that is also rampant. Anywhere you go, whether you enter into a bank, whether you enter into a shop, there will be someone standing with a bottle of sanitizer sprinkling on your hand. And you never know whether it is a good product, whether it is a reliable product, because hundreds of household businesses started making sanitizers at home and selling it with some brands. So unbranded sanitizers entered into market, which is not really that efficient. Repeated hand wash with soap. So all these ultimately keeps your hand dry, that kills your normal flora. And there are reports that excessive use of these alcohol-based sanitizers ultimately kills your dendritic cells, which are there deep inside your skin. And ultimately these dendritic cells were your immune soldiers. So is there any alternative option for that? So that's how a new opportunity comes. Is there any natural product-based sanitizer? But again, the efficacy is questionable. Every day you will see some news article that someone come up with some natural product-based solutions, but who is going to test it? Because we don't have a real in vitro model or in vivo model or biomarker, as I told in the earlier slides. So there is a lot more requirement in that. So that at least we can prove or we can validate the claim. So, and that's the reason the sanitizing devices came into picture. In the first or second month, you must have seen that tunnels were made, right? You just walk through the tunnel and you will be showered with some chemical, and you are claimed that you are clean, right? So they were sprinkling sodium hypochlorite, and few of them they sprinkle even isopropyl alcohol, so which is not advisable, because sodium hypochlorite is not for human use. It is for discarding the waste. So this sodium hypochlorite will react with your skin, and that will create other issues. So then again opportunity came. Any alternate to this? So then came the alcohol-free sanitizing device. I will discuss that later on in my talk, because we developed one alcohol-free sanitizing device with the support from GUSAC. Now you will see a lot of equipments or the surface cleaning claims are coming out. One of them is UV irradiation. So you will find small boxes or small tunnels, and you put your luggage or you put your mobile or your wallet, and they will irradiate with UV, and ultimately it is clean. So the issue with UV is that the higher wavelength UV are hazardous. So say UV 254 is carcinogenic, so genotoxic. So any eventual irradiation to the user may create issues, right? So now there is a scope of improvement in making these devices which are safer. Even in the UV spectra, there are specific wavelength of UV which is least toxic to human, but they are effective against microbes. So you have to find out those device sources, and we have to make it indigenously, because now our Honorable Prime Minister is promoting Swanirbar Bharat, so that make in India, we can produce everything in India, because LEDs were not available or they were not manufactured in India. Now, this is the time we have to think on these aspects also. So other preventive measures is be active, exercise, yoga, pranayam, meditation. Avoid panic and anxiety, because anxiety also is a form of stress that kills your immunity. Proper diet and nutrition, is a preventive measure. Now let's go to the treatment part. As I discussed earlier, in the allopathy, they came up with hydroxychloroquine, remdesivir, and loperamide with interferon that where they were having, because either it was an antiviral drug or it was anti-HIV drug. And since it is a virus, they thought that it will at least treat something or to some extent. And that's how the whole drug repurposing area came into picture. And there was a lot of opportunity for bioinformatician in this. Here, with the help of molecular modeling, molecular docking, molecular dynamics studies, we can repurpose the FDA approved drugs against the various targets that I discussed in my earlier slide, the red T. So there were around 10 to 11 various junctions where we can target these drugs. So there's a area for a bioinformatician to do that. Second one is patient support. Say vital monitoring in ICU is a costly affair, as well as we have limited number of seats in the ICU. So what if tomorrow there are 1 lakh patients in Ahmedabad and we don't have ventilators, we don't have ICU facility, we don't have medical facilities to cater their need. So, and that's how the cost-effective ventilator came into picture. But unfortunately, the disease, which initially it was presumed will be supported by ventilator, is not recommended nowadays, because this is not the severe respiratory distress type of disease in the lung, but it is hemostasis that is happening in the lung capillaries. And that's the way the whole blood supply is being retarded and patient enters into hypoxia. So now these ventilators are not a lucrative business. Role of macro and micronutrients, that means opportunity for food and nutritionist. So they can design a cocktail of supplements which is beneficial for this type of clinical conditions. The last hope is vaccine. We all are waiting for that, but still it is expected not before 18 to say 24 months. So it is way far from us. But still, even if it comes, efficacy is questionable. Now, if you look at the COVID vaccines, the vaccines under active trials, they rely mostly on different viruses or viral parts. Say a whole virus vaccine, which is either attenuated or inactivated by various methods, can be directly injected into the body so that body acts on that. But the disadvantage is that it requires extensive safety testing. I think Sinovac Biotech is working on that. And that's the reason people thought of other modalities, say viral vector vaccines. Here, a different virus, which is less lethal or which is genetically engineered to be non-pathogenic, so adenoviruses, it can take the coronavirus protein inside the body, and it can be replicative or non-replicative. So that depends, and ultimately these acts as a carrier. There are certain nucleic acid vaccines where synthetic RNA is inserted into human cells, and which then churns out the copies of viral spike proteins. So this nucleic acid is not, it is customized, and it is not having the pathogenic islands of the genome, but it is only having the structural proteins, so that when it replicates, only the structural proteins are released into the body, and based on that, the body will respond to the viruses. There are protein-based vaccines. So fragments of proteins or the protein cells that mimic the coronavirus outer coat can also be used. There was a assumption that possibly a BCG vaccination, which majority of Indians have taken, has some role in protection against and COVID. And still, That is a guess only, but I think it is working. So people says that whether COVID-19 vaccine is a hype or hope. As I told, it can be protein, it can be DNA, it can be RNA, or it can be modified virus. So various agencies are working in this. Moderna has released recently, 2 or 3 days back, that its initial clinical trial of mRNA vaccine is going well. But the efficacy is questionable because of high mutation rate in this RNA. So majority of these coding regions are getting extensively mutated, so that when the vaccine comes, and at that time, the strain, what is the mRNA constitutions, will it be efficiently acting on those viruses? Second thing is long-term immunity is not feasible. So if you look at even the influenza vaccine, every year or two, you have to take that. So it is not a lifelong vaccine, right? So now GU has ventured into a small antigenic peptide-based vaccine along with Neo7 USA. So this is put up as a Indo-US proposal for this COVID call. And here what we did is the whole viral protein coding regions, or the viral protein segments, they were fragmented into smaller peptides, and the most antigenic site in that peptide are taken as if it is compatible to MSC1 or MSC2 antigenic presenting cells. So once it stimulates the antigenic presenting cell, those antigen will be presented to CD4 or CD8, and ultimately that will help in developing immunity against that specific antigens. So these are not the complete epitope, but this is the part of that. And as it is required that around 9 to 15 amino acid long stretch antigenic peptides, so it can be synthetically made. One more recent advancement was in the field of stem cell therapy. So stem cell therapy in COVID-19, you can see there are a lot more paper that came out in the last month. Mesenchymal stem cell therapy in COVID-19. So here what they did, they injected mesenchymal stem cells intravenously, and these mesenchymal stem cells, they colonized in the lung epithelium, and there they rejuvenated the lung epithelial, and they helped in reviving the lung function. Some of them used the exosomes derived from this mesenchymal cell culture, and they were directly spreaded onto the lung epithelial lining, and they reclaim the pulmonary microenvironment, prevent pulmonary fibrosis, and they cure lung dysfunction. So these are scattered claims, but this need to be extensively validated through clinical trials. So if I have to say some do's and don'ts in this, I can say exercise regularly, self-hygiene, social distancing, balanced diet, and macro and micronutrients. Maintain your sleep and wake cycle for our circadian rhythm, and don't take medicines prescribed on internet without any guidance. So avoid self-medication. And it is also mentioned in Ayurveda that ahar, vihar, and visram. So that keeps us healthy and safe. There are many myths circulating around the internet, and that's the reason WHO passed on several such myths. The spraying alcohol or chlorine all over your body kill the new coronavirus. So as I told, these tunnels are now banned in many places. Viruses doesn't survive in warm weather or in the sun. Then they should not be there when the temperature in Ahmedabad is 46 degrees. Gargling warm salt water can help prevent COVID-19. So this has not been proven clinically that these respiratory viruses are not impacted by salt water. And possibly, I think some of the Ayush people, they are trying to validate this. One myth is that if you can hold your breath for 10 seconds, you are healthy. So according to the experts, the 10-second test being shared online would not be reliable way to determine whether the patient has virus or not, but it can definitely tell whether your lung function is still better, right? So when you can do this, that means you have got a severe lung injury. In this time period of last 2 months, there were a lot of funding calls from various funding agencies. So that was really a good chance for all the innovators or startups to venture into. There were calls from ICMR, DBT, CSR, DST, Ayush. Even GUSEC call is still open for breakthrough innovation, and this is the details. So anyone, students or any individuals with some good ideas, which I discussed in the last few slides, may apply this online, and it is still open till 31st of May. Which cost call is expected possibly in this week or in the next week. So as Professor Jain said, I'm associated with GUSEC also in the board of director, and that's the reason I'm showing this. That GUSEC has a vision that it's an early stage, 0 day, 0 cost. That means for incubation, you don't require to pay anything, and you can be incubated there. A startup support system with a mission to convert 1% of the 4 lakh students of Gujarat University ecosystem into entrepreneurs. So if we succeed in this, then we will have a lot of entrepreneurs and innovators, which can build the economic and growth value from within a university ecosystem. GUSEC's vision is to give an accelerated platform to idea level entrepreneurs and support them with all necessary resources and mentorship to build the real value from their current idea. Now, this is our time to see what are the innovations taken or undertaken at GU or GUSEC during the lockdown period. The first one is we developed an alcohol-free sanitizer. So this project is completed, and this has already been demonstrated in media, and our Honorable Minister of Education, he was there physically. Now, what we have proposed is UVC based room, cabinet, or PPE sanitizing for portable devices for personal belonging. And here the uniqueness of this UVC is that it is based on wavelength 222. And at this wavelength, it is least toxic to human cells, but it is effective against all the pathogens. Development of nosode. So this is one of the unique aspect in homeopathy, and this is proposed in collaboration with the Homeopathy Foundation, Life Force Foundation from Mumbai, an Innovative Thought Forum from Ahmedabad, along with GU. It has been proposed to Ayush. So here it is also similar to a vaccine which is there in allopathy. So what they do, they take inactivated form of the viruses, and they do dilutions in alcohol, and with every dilution, it is being given 15 to 16 strokes, and that they call suctions. And when they reaches up to say 12x or 30x, virtually there will be no traces of the intact viruses in them. But these type of nosodes with other illnesses are already there a few years back, and they have got proven efficiency. So this will be in market in a very near future. Development of peptide-based vaccines with Neo7 Logics USA, as I told, it was proposed in a Indo-US collaborative proposal. Point of care device for COVID diagnosis. So this is also proposed to DST, SERB, along with some collaborators, because you won't have all the expertise as well as all the facility. So it is always advisable to hold the hand of a collaborator who has got expertise. So here we are trying to develop a point of care device not based on the antibody type of rapid care device that I discussed earlier, IgG, IgM, because the main issue with IgG-IgM based device, that you must have read, that the efficiency of the sensitivity was somewhere between 40 to 60%. Why? Because many times these antibodies, if they are not properly aligned, they will not bind to the antigen. Or since these are antibodies and these are protein, intact protein, their stability becomes an issue when you take them to the field. So what we did, we worked on aptamer-based binding. So these aptamers are synthetic peptides or nucleic acid fragments of around 20 mers. And a specific aptamer, so millions of aptamers were screened against the target in silico, and the best binding aptamers were then printed on these devices, so that when you pass on the antigen or the antibody, it can bind efficiently to those and it can detect them. So that's why we overcome the antibody-based devices. Special coating material and special fabric for PPE. This is also ongoing. And here, the silver nanoparticle-based spraying on the various PPE garments have been proposed to different agencies. Polyherbal formulation, as I told, Immunize is already proposed to ICMR and Ayush, government of Gujarat. And this clinical trial will begin soon. This was the layout diagram of our alcohol-free sanitization product, where we used hydrogen peroxide, ozone, and the cold atmospheric plasma discharge unit, which ultimately formed a mist of highly charged ions. And when they go and hit the target, they will inactivate all biological agents present there. This was also validated in the lab by the culture methods, but this was trial and tested on bacteria currently, and now it will be tested on a setup where there is BSL-3 or 4, and they can work on live viruses. Different models of that can be used as per the need, whether it is specifically for hand and face sanitization, or it is whole body sanitization. So if it is for OT or a lab, it can be a chamber like this for whole body sanitization. And if it is at entry and exit point of various outlets, it can be for face and hand sanitization. So for this, we mentored 3 students of engineering, and as a biologist, physicist, and engineering students. So this is a collaboration. And there is no harm in collaborating with anyone, whether he is younger to you, whether he is older to you, that's fine. And during this lockdown period, with a collaborative effort, we could do some bioinformatics-based publications, like identification of potential binders of SARS-CoV-2 spike proteins, and the hemagglutinin esterase, mainly through molecular binding, simulation, and pre-energy calculations. So this was approved in the last week. So for the young budding mind, I would say dare to think out of the box, be innovative, integrated, and do think interdisciplinary. Some collaboration with a heterogeneous collaboration with different experts. So the take-home message can be: age is just a number. Don't fear in collaborating with experts. Be vigilant and empathetic, so that what is happening around you, and is there a innovation possible there? Can you intervene? Can you do something for that? So you have to be empathetic for that. Identify the real need. So what is the real need? So I saw once that someone is preparing a face mask with some biological detection agents using CRISPR-Cas and all those complicated mechanism. But that will tell you, once your mask color changes, that means it has been exposed. The idea is good, but is it really feasible on field? Because that mask will cost you somewhere around 1,000 rupees. So identify the real need and look for an effective and affordable solution. act promptly, because opportunity never keeps on knocking the door again and again. So thank you all for patient hearing. And I would request you, don't be fearful, but definitely be careful. Sarve bhavantu sukhina, sarve santu niramaya. So that is